Yes, hi everyone. I'm Kevin. I'm a recent appointee within the School of Agriculture and Food Science here, and I've been invited to contribute to this seminar along with the much more exciting Laurent Frantz, who I'll kind of introduce later on, and I'll give you a bit of an introduction to our the field that we both work in, and so with that, I'll just host disabled participant screen sharing. I think I need to be able to share my screen. Could I be, Gary, could I be made a host?
I'm able to share my screen, Kevin, so I think you should be able to try.
I can now. Thanks. Give me 1 second. And people can see that?
Oh, good.
Yeah, great. So I'll tell you a little bit about what paleogenomics can tell us about livestock and their pathogens, and how we can kind of learn how that the past shapes our present and our future as well. So just kind of like to set the stage, what's the origin of agriculture? Where does our food production systems actually originate in? And we typically when we talk about this, we typically refer to the Neolithic period. So this is the new Stone Age, and when people began to practice, they were maybe sedentary already. They actually began to practice both kind of crop production, but also animal management. And now there was this kind of occurred in different places around the globe at different times, but typically I personally focus on Southwest Asia, and roughly at a time period around 10,000 years ago. Now, how can we actually study this sorry, I'll get onto the genomes later. And just to kind of talk a bit more about Southwest Asia. So, and if famously there was around 4 species domesticated, again, roughly around 10,000 years ago within Southwest Asia. So this is kind of across eastern Turkey, northern Iraq, and western Iran. And those species are goats, pigs, cattle, and sheep. Now, other species were domesticated at other times in different regions. And famously, pigs were also domesticated in China, maybe I think 6,000 years ago or 8,000 years ago. Laurent can correct me on that as well. But this is a kind of a core region within Southwest Asia where we've had several different species domesticated, roughly around the same time. Now we can, from remains of these animals, so bone remains, teeth, particular parts of the skeleton, we can actually recover and extract DNA, and using that DNA, we can reconstruct their genomes and kind of infer how these past animal populations relate to those today, and therefore understand their domestication process. So to do this, first we have to basically wear boiler suits, personal protection equipment to stop us from contaminating the material we're working with, because the DNA that does preserve is preserved at very low quantities. And I'll just give you an idea of the kind of workflow that I would go through. So I might start with a bone such as this in the top left. It's a petrous bone. It's part of the skull that surrounds the inner part of the ear. And then with a saw, effectively like a hand drill, I'd cut out a chunk of the densest part of this material, grind it to a fine powder with a mix mill. Take about 100 milligrams of the bone powder, so not much. In protocols we use now, we also give it a very light wash with dilute bleach, which does wonders for removing kind of exogenous contaminant molecules. Then we just digest the bone matrix, which is still present, using EDTA and Protease K. So like a really simple kind of extraction buffer that just runs for a day or two. And then this releases tiny fragmented parts of DNA, which we can then do kind of modern shotgun sequencing. And from this sequencing, we can then reconstruct genomes. So my own research has focused on the humble goat, Capra hircus. While goat today isn't the most important production species. It was much more important in the ancient past, particularly in the Neolithic period, maybe parts of South Europe. So the domestic goat was, has a deep past with us. So this is a mud brick that should date to around 10,000 years ago. So if you can see, there's actually about 3 different hoof imprints in this mud brick. So this was, and this comes from a settlement known as Ganj Dareh in Western Iran. So again, about 10,000 years. So these early herders would have literally been, setting the stones of their houses, their settlements, and then their herded goats would have been wandering through and even just stepping on the building. So clearly, like, go back far enough in time, and these animals they're intimately related to kind of early kind of herding societies. And so they were domesticated from Capra aegagrus, also known as the bezoar, which are phenotypically quite a bit different. They're bigger. They have larger horns. They show less coat color variation. So trying to understand the genetic process leading from the bezoar to the domestic goat was kind of part of my PhD focus and something I still work on. To give you an idea of where this occurred, so this in blue is a distribution of where the wild bezoar is found today. It's a bit of a fragmented distribution because their habitat has been on the decline for the last 10,000 years. In X are some of the earliest sites where there's evidence of goat management. And then on the right, kind of in Western Iran, is Ganj Dareh, the site I mentioned earlier, which has quite robust evidence for goat management. So what does this evidence look like? So about 10,000 years ago, the goat from this site saw really strong sexual kind of sex-specific survival patterns. Basically, about the age of 12 months, you get differential survival from males and females. There's a sharp increase in the number of males dying effectively, being slaughtered, whereas females are allowed to grow to an older age. So it's thought that this is a early managed herd. You're keeping the breeding females at a higher proportion than the breeding males, such that you're maximizing your returns. You're getting meat from your males and you're getting offspring from your females. You're managing a herd. Now, the interesting thing is that this is a herd of morphologically wild animals. We still have the bones which represent these goat. We have their horn cores, and we can see that they aren't actually distinguishable from wild bezoar. So they're managed, but they're morphologically wild, which makes this a kind of intriguing kind of population to kind of assess their genetic makeup. So this is something we've actually done in a few years ago in a paper published in 2021. We managed to extract about 12 genomes from this really early herding site. So this is a tree which is relating both the genomes from this really early herding site, Ganj Dareh, to other ancient goat genomes, to modern goat genomes, and then also to wild bezoar genomes. So the bezoar genomes fall at the very bottom of the tree. And what we see is that our genomes from Ganj Dareh, so this is the earliest Neolithic herds with the big red arrow pointing at it. So they are actually at the very base of all later goat kind of variation. So basically all domestic goat fall in their own group, but at the very base of that group is the Ganj Dareh goat. So these early herded goat are kind of very much related to the ancestry of goat today. They're genetically quite related to where all of modern goat ancestry comes from. And then we actually also picked up some more divergent individuals. So that's that box in orange, which appear to be wild goat. They appear to have more kind of wild genetic affinity, which suggests that these early herders were also still hunting animals. And you can see that in well, not just animals, they were still hunting goat while they herded them. And you can actually see that in the kind of the mix of animals, kind of animal remains at this site. There's like foxes and birds and so on, which have been butchered. So clearly these are early, goat herders who are also supplementing their diet to some degree by still hunting different kinds of animals, including wild goat. So this early herding impacted the genomes of these animals in different ways. One thing we think that we pick up in these early goat herds is indications of inbreeding. So each of these plots here is a different chromosome, and how high a point is on the y-axis is how much kind of genetic diversity we see between two copies of the chromosome that a particular ancient goat carries. And what we see is there's in our different ancient goat from this site, we see long regions of chromosomes which don't actually show any genetic diversity between the two copies of a particular chromosome. So this is called a run of homozygosity because the two copies they're homozygous. They show no variation. And so this typically happens when you have mating between relatives. Okay, particularly the longer the run, the more recent the inbreeding event was. So we might actually think that if you don't have a wild population which is kind of breeding as it likes, and instead you have humans which are controlling the survival of certain individuals, you get more more males being killed off at the age of roughly between 12 months and 24 months. You're reducing your breeding population. You're creating the circumstances for more inbreeding. So this is kind of the genetic stamp of early husbandry in a sense. It mightn't have been kind of goal orientated in the sense of changing the genetic makeup of these goats, but there was a genetic impact already of these early of this early management. Now, there's other ways we can look at the impact that humans have on the makeup of domestic populations. So another way is to look at selection. So there are genetic variants which have risen in frequency due to being more favorable. So that could be due to increased survival versus some pathogen or specific selection by humans. So we did this in an earlier paper comparing domestic goats from around 8,000 years ago to wild goats today. And what we were trying to look at is regions in the genome which show high genetic distance. So they're very different from the wild goat genome, but they also show lower genetic diversity. So only a kind of a small amount of genetic diversity is present in these domestic goats in these particular regions. And so what we're trying to do here is find regions of the genome which have underwent fairly strong selection in Neolithic goat genomes. And by making this comparison, we were actually able to pull out regions of the genome which overlapped genes which have like quite a tangible link to kind of phenotypes. So how the makeup of an animal. So one of the strongest results we had was actually from pigmentation related genes. So these are genes, so kind of famously KIT and KIT-LG are classic co-color genes in domestic animals. Usually they're associated with white spotting, actually. And so we saw this was actually recurrent. We saw this recurrently in different populations that we looked at. We saw the KIT and KIT-LG genes showing this signature of selection, which might suggest that early farmers were, whether knowingly or unknowingly, they were showing some kind of like favoritism towards different co-colors compared to their wild progenitors. We also saw signals of selection in genes linked to milk yield, mammary development, other production traits, then also cytotoxicity, so the ability to break down potential toxins. And this specific gene, it's a cytochrome gene is actually associated with fungal toxins, which have been suggested to be linked to kind of grain storage. So it might be an adaptation to essentially to foddering or to us feeding animals grain supplies. Now, more recently, I've begun to work with genomes from domestic sheep, so which are, I would say, a bit more of a kind of an important production animal today, anyway. Now, they were domesticated from the Asiatic mouflon, again, quite a bit different from their domestic relative today. Just to locate you again, we're talking of the same general region, which shows the earliest evidence of sheep management. It's about 10 to 11,000 years ago in Southwest Asia, maybe a bit more, pushing a bit more west into Turkey as well. Now, this is unpublished work, so I just ask not to be shared too broadly, but we've generated a pretty large data set of 118 ancient sheep genomes. We've done various different kinds of analyses, but I think it'd be interesting just to draw one which has a parallel to the results in goat. So we did a similar analysis for selection signals, not the exact same kind of analysis. This time we were comparing 3 populations at once, basically comparing a wild population to populations about 8,000 years ago, so during the Neolithic period, but then also comparing more recent populations. So when we look at populations about 8,000 years ago during the Neolithic, we saw quite strong selection signals across a wide range of coat color related genes. Well, this plot is a little bit out of date. We do see just a broad kind of range of different pigmentation related genes, which show an excess of this selection signal compared to what we might expect from kind of a comparable set of genes. Which again, it's actually because of the concurrence between 2 different species, it does suggest that in general, early herders were favoring either a diversity of colors of coat colors, maybe to kind of denote ownership within shared community herds. So when we make the same kind of analysis for later populations, so those are about 4,000 to 1,000 years ago, we don't see anything in pigmentation. We do see signals in other interesting production-related traits. So we get a really big, strong selection in horns in this kind of RXFP2 gene, which is very famously associated with hornedness or polledness. It's known as the polled locus. So it's variation there affects whether an animal does or doesn't have horns, or whether there's sex-specific hornedness in a particular breed. And we also see, I'd say, weaker kind of selected signals in different production-related traits, so wool and milk. There's fairly robust evidence that people were drinking sheep's milk during this kind of time period, the Bronze Age to the more recent time periods. And this is also around when wool becomes a kind of a production commodity across Western Eurasia. So we're getting kind of a weaker signal there that of this genetic basis of this production commodity. Now, it's not just livestock which are shaped by kind of human activity, it's also the pathogens which affect them. Now, in a lot of these early Neolithic sites, there's a high frequency of basically baby sheep and goat bones, fetal bones, perinatal bones. It's been suggested of sheep and goat, if it wasn't clear. Now, it's been suggested that either stress or infection is the cause of the high frequency of these bones in these early herding settlements. Now, I've been lucky enough to work in an SFI, pathways project, screening material like this and other bones for traces of pathogens, traces of ancient pathogens. Now, I'll tell you a little about one we found, which is quite well known. This is Brucella. So Brucella is it's a zoonotic pathogen which primarily affects The most famous species affects livestock. Now, in both animals and humans, it can cause abortion, like spontaneous induced abortion through infection of trophoblast. It also causes illness in other ways. It causes this insulating fever, and then it's transmitted within herds via the aborted material, and then humans can pick it up by drinking unpasteurized milk or kind of just too close proximity, physical contact with infected animals. Now, different species of Brucella infect different host species. So, for example, Brucella abortus infects mostly cattle. Brucella melitensis affects sheep and goat. But there's a bit of promiscuity in the species that infect. The one which infects humans most frequently is Brucella melitensis, the sheep goat infecting one. So we've managed to pick up an 8,000-year-old Brucella genome from one of the sheep, one of the 118 sheep that I just mentioned from our sheep analysis. One of them also carried a Brucella genome. So when we ask, how does this Brucella genome relate to other Brucella species, what we find is that it's a Brucella melitensis, so it's the species we think it should be because it was found in a sheep, but it's right at the base of Brucella melitensis variation. So it's relatively recent after Brucella melitensis has evolved from a common ancestor with the cattle infecting Brucella abortus. And we can actually put a distinct time point on that. And because we've dated the bone from which we got the pathogen, we can use that to calibrate the genome tree which relates our ancient Brucella to Brucella melitensis today, and then also the relative Brucella abortus. And with that, we can kind of put a point or a range estimate on when melitensis evolved from the common ancestor with abortus, which puts it to around 7,800 years ago, which is right in the middle of the kind of the Southwest Asian Neolithic period, which suggests that this livestock pathogen did actually evolve and diversified within the context of the development of animal farming, effectively. So, and that's now has a consequence for both animal health and then also human health, given that this is, I think, the most common bacterial zoonosis today, Brucella melitensis, affecting something like 3 million people every year. And so I think this is a good time to pass it on to Laurent, who will give a very exciting overview of the research that he does. So Laurent's a professor at LMU, Ludwig-Maximilians-Universität in Munich. His work, I think it's the animal paleogenomics team, focuses on a wide range of domestic species: pigs, cattle, chickens, sheep, wild species as well. And I'm looking at them in terms of their domestication history, their contribution to modern breeds, their role in conservation biology, and also kind of epidemiology. So looking at kind of the pathogens which might affect them as well. And he's been involved in some very exciting and innovative studies recently that I'm sure he's going to tell you all about now.
Thank you, Kevin. Okay, let me just try to share my screen. Find the right thing. There we go. Can you see my screen? Yeah?
Yeah, all good.
Right. Great. Thank you, Kevin, for this intro. There's lots of stuff I can then skip in these slides. So it should be, let me just remove this. There we go. And yeah, thank you very much for inviting me to this. This is very exciting to talk to you guys about what we're doing here in Munich. So I am based here in Munich and at Queen Mary in London, although I'm mostly physically in Munich, but I do travel to London quite often. So as Kevin mentioned, our group is sort of generally interested in animal genetics, and what really drives us is to understand the impact of human activities on animals. We are interested in sort of long-term processes such as animal domestication. We're also interested in processes that happen due to hunting, how animals are being shaped, the genomes are being shaped by hunting practices, and by domestication processes that happened thousands of years ago, like Kevin talked about. We are also really interested in understanding how the genomes of animals is affected by things we're doing today or in the last 100 years, breeding and the exposure to pathogens more recently. So I'll talk a little bit more about this later. And another thing that my lab is quite interested in is trying to understand not just the domestic side of animals, but also what's happening in the wild. So we're really interested in understanding the impact of human activities on wild animals. So understanding why do some species go extinct, why some don't, and trying to provide some feedback, some information to conservation biologists to try to improve the survival prospects of some endangered species. And a lot of what we do, as Kevin mentioned, is genomics. We study the genomes to try to understand the evolutionary processes that took place in the last, let's say roughly 100,000 years, but mostly in the last 10,000 years. But rather than studying these processes by just looking at the genomes of animals living today, we are working with genomes that have time-stamped information, so we can generate genomes from archaeological sites that provide us with information on how these evolutionary processes take place in real time, rather than inferring information directly from modern genomes. So it really offers us a possibility to sort of travel back in time and see and actually observe the changes that happen in these genomes and try to link them with specific changes in human activities. So one of the big things that our lab is working on at the moment is conservation biology. We have a couple of projects at the moment where we're looking at how deforestation is affecting the genome of animals in places like island Southeast Asia, and we work with people doing spatial modeling. And we work with historical genomes, so we compare the genomes of animals 100 years ago to the genome of animals today, and see where are the places in the islands, on these islands, where they've lost most of their genetic diversity, and to try to predict where are the places where they are the most endangered. And then we can combine this information with information about spatial models like forest covers. We also work a lot in the biology of extinction. It's trying to understand what drives extinction of species. We work a lot with canids, so because we work a lot, the lab works a lot on dogs, that allows us also to work on wolves and related species. So we work a lot in coyotes and canids and wolves in North America. I'm particularly interested in what happened when we killed about millions of coyotes and wolves in North America in the 50s and in the first part of the 20th century. And so that leads to rampant hybridization between different species, the coyote and the gray wolf, and also the local extinctions of species. And so we can go back in time and sequence genome that happened just slightly before this to try to understand how the biogeography had changed on the continent. We also work on cats recently. I worked on the domestic cat and the wild cats in Scotland, and trying to understand how basically by persecuting domestic wild cats in Scotland, so as their population reduces, we've basically led to a possibility of hybridization between wild and domestic cats, and sort of trying to understand how we can prevent this in the future using historical and modern genomes. We have we're lucky to work with a lot of archaeologists as well. So we work on questions related to how people have moved in the past. So looking at migration routes, at the moment we work on pigs in Polynesia and island Southeast Asia, for example, to try to understand what routes did people take when they brought farming into island Southeast Asia and migrated into Polynesia. And so we can track the genome of the pigs they brought with them to try to understand these processes. We worked a lot on dogs as well in the lab in North America to try to understand the peopling of North America, but through the dogs they brought with them. But probably the most important area that our lab is interested in is animal domestication. So we are interested in understanding the where and when, and how many times specific species. So like Kevin said, our lab is really interested in dogs, pigs, and chickens. We do work a little bit on cattle and sheep, but mostly pigs, dogs, and chickens. And we're really interested in understanding what happened to the genome during domestication, and trying to understand the impact of industrial farming on the genome of the animals, but also of their pathogens. So because I guess this is more of an audience that you guys are more interested probably in the sort of maybe more recent stuff, and that what Kevin has talked about is sort of really early domestication time frame. I thought I will talk about more about recent evolutionary process that happens in species that we are farming today extensively. And so what I want to take you to is our recent work on chickens. So I'm going to try to give you an overview of the evolutionary of chickens, going back in time to the early domestication process. But really, what I want to show you is how the evolution of chickens had really sort of picked up in the last 50 years or last 60 years, really after the Second World War. So chickens that we have today in Europe were first domesticated in 4,000 years ago, give or take, so quite much later than the species that Kevin was talking about, the goats, the sheep, the cattle, the pigs. These guys are domesticated maybe 8,000, 9,000, 10,000 years ago. Dogs go back to maybe 20,000 years. So chickens is a really recent thing, relatively recent, similar time almost as horses. They were domesticated from a species of wild jungle fowl, Gallus padiceus, which is living today in between Myanmar and all the way into the border between Vietnam and Thailand and Cambodia. So from this time frame, about 4,000 years, the chickens started being spread across the world by people. And it took about 3,000 years to arrive in Europe. We start seeing chickens popping up in archaeological sites from the Levant, so in present-day, Israel, Palestine, and Lebanon, in the Hellenistic period, so the Greek period, where you start seeing chickens being more and more present, and so we think chickens get, about 1,000 years later after the domestication start arriving in Europe. So about 3,000 years ago. For most of the time at which chickens were in Europe, for most of the last 2,000 years or 3,000 years of chicken evolution in Europe, they were at really low. The low consumption of chickens generally is in Europe, right? So we have very few chickens at archaeological sites. So for example, this is a graph here that shows the proportion of chickens that you find at archaeological sites in different time periods from the UK. And so here, this is the percentage of chickens compared to other species. And you can see here, basically the 90% of the assemblages, so the archaeological sites from the Iron Age, in the UK, which is just before the Roman Age here, less than 1% chickens. Very few archaeological sites have a lot of chickens. Most of them have very few chickens. The majority of animal bones that you find are from all the other species that Kevin talked about. But so this shows that people were not eating chickens that much in the UK, at least during the Iron Age and Roman time. And a lot of why this bird was used for was probably more, cockfighting, religious purpose. Some Romans had some weird, a really weird sort of religious practices with chickens, and also trying to predict the outcome of war and things like this through divination that based on chickens. So chicken were really not necessarily seen as a source of food for a long time in Europe. And we see the same thing in the Middle Ages. This is again the proportion of chicken remains in sites in Germany and in the UK. And so you can see in the Middle Ages, it doesn't really make a huge proportion, maybe 10% of the sites well, 10% of the chicken remains in sites of the 12th to the 14th century. In England and in Germany, even less. So we can, what this is all pointing out is that really for most of the evolutionary history of chickens in Europe, it was a really marginal source of food. This is until really first part of the 20th century, but really post-World War II. This was driven a lot by, basically policies in the US and in Europe that tried to produce, cheap, healthy source of proteins for people. And so at this point, chickens started taking over dramatically cattle and pig consumption since the 1960s. And you can see in this graph that the proportion of chickens is increasing dramatically. It's increasing much faster than the consumption of cattle and pigs over this time period. It's not just the consumption of chicken, it's also the chicken themselves have dramatically evolved over that time period. So much so that their phenotype has evolved dramatically. So here we have on top here we have the picture of a chicken line that was basically left alone since the 1950s. So people haven't selected for this line; they just let it to mate the way it wants. So let it evolve without human, without much directed breeding. And so you can see, at 85 days, this is the size of a chicken that hasn't been selected by people. And then this is the same line that was basically selected from 1957 to 2001 for bigger breast. And you can see what happened to this animal here in just 85 days. So dramatic changes in the biology of that species, driven by this increase in demand for chicken meats worldwide, really worldwide, but really also probably more even in the West for the majority of the second half of the 20th century. And not only the phenotype of these individuals have changed, but also the way that we are, the way that we are raising them. The husbandry practices have changed dramatically. So this is actually quite good. Chickens have actually access to some space. They can roam on the ground. This is not always the case, although in Europe we don't have chickens that of soil, but in the US they do this. So some chickens are so crowded in cages they never even see the ground. But what I want you to think about here is in terms of the kind of infectious disease aspects of a husbandry practice like this, cramping together thousands and thousands of birds in a species that essentially is almost solitary in the wild, right? So we have these jungle fowl that live almost solitarily in the wild, or they're not really living in groups, hosting to basically a few backyard chickens that people were keeping in for most of the last few thousand years to this in just a few hundred years, or maybe in just a few decades. And so this has led also to a dramatic evolutionary dynamic between the chicken immune system and also the pathogens that they were affected by. And basically one outcome of this process is the evolution of a disease that's called Marek disease, which is basically a virus that affects most industrial poultry farms today in the world. So the waves of infection all across the world since the second half of the 20th century. It basically requires near universal vaccination because unvaccinated chickens will die, about 100% rate. And it's estimated it costs more than $1 billion a year, basically, to vaccinate all these chickens. And also due to the loss of birds that results from infections across the world. And then it wasn't always like this in the 20th century, since this virus was first described by Marek. And at the time, it was the symptoms that were mostly seen was this flaccid paralysis, which is basically chickens were having transient paralysis for I don't know how long, but just a few minutes, and then could go back to their normal life. And this was really what the virus was driving in terms of symptoms. And this symptoms have dramatically evolved in the second half of the 20th century, and particularly since the 1960s. Coincidentally with the introduction of vaccines as well for this disease. So now it results in pretty horrible symptoms like edemas and lymphomas, and it'll basically kill the chicken fairly quickly if it's not vaccinated. And the other problem with Marek, and what makes it so well adapted to the chicken industry, is that its life cycle means that it is transmitted through feather dust. So it gets into feather follicles here. If you're looking at this side here, it gets to feather follicles, replicates to feather follicles, and then it's shedding in the air through the feather dust here, and then it's inhaled by chickens through the lungs. So you can imagine that if you go back to these sorts of husbandry conditions, it will spread like wildfire very, very rapidly. And so as soon as you have one chicken that's infected, then everybody is infected a few days afterwards. So it really is the virus that really well adapted to modern husbandry practices. There is also an issue with the vaccine itself, because the industry needs to keep costs down, obviously, to keep the price of meat low. They need to use, they're using vaccines that are maybe not, the vaccine basically don't provide any resistance to the disease in a sense. It doesn't prevent transmission. It basically delays the onsets of the symptoms. So if a chicken flock is affected by Marek and that's vaccinated, they won't die very quickly. They will die only. They would die eventually, but they won't die very quickly. So they will be able to survive to slaughter age. So this is not as much of a economic loss from the farmer. But the problem with these sorts of vaccines and this imperfect vaccine is that it is possible that it actually leads to selection of more virulent type. And this was modeled by a Venus group here in the Purbright Institute in the UK, which suggests that actually this might be the case, that by with this leaky vaccine, as they call it, or this imperfection vaccination can lead to this selection of virulent strains. And so maybe this is what we are doing here. So we're providing a huge amount of possibility to spread very quickly through the population, and even maybe boosting the evolutionary process even more through this administration of a leaky vaccine. And so the big questions really that we have about Marek disease is really, for how long has been circulated in chickens? Is it something that has been around for a very long time and that really was driven to becoming such a beast very recently? And the other question that we had is, what is the molecular basis for its virulence? What drives the virulence of this virus? And one of the big problems to answer these two questions is that we don't have really any information about how the genomes of this virus looked like before basically the 1970s, which is the first few samples that we have that have been kept since then, that have been sequenced. Before this, we don't have any information. So we don't have any information about the genetics of Marek disease prior to really the onset of it becoming a very virulent virus. And so it's really hard to figure out what drives it, and what makes it virulent if you don't have any comparative data to assess where the changes have had taken place in the genome. And so this is where ancient DNA comes in. And as Kevin showed you, once you're sequencing DNA from bones, you don't only sequence the DNA from the host, from the animal that died, but also from all sorts of other commensal and pathogenic species that were living in with the organism that died. And so we can sequence pathogen genomes from this. So it's possible with Marek as well because it does spend time, a lot of time in the bone marrow. So it leaves traces. So we were able to actually isolate DNA from this DNA virus from chicken bones from archaeological record. So this is an overview of the samples where we've been able to obtain Marek disease from. So you can see that it's quite a few places across Europe. In X here is the coverage of the genome. So you can see the genomes are really low coverage in general. It's quite hard to get much data. I won't go into detail about how we generate this data, but if you have any questions, I'm very happy to answer that. But as you can see, we can push back the time of of Marek to about 1,000 years before present, so to 1,000 years AD or CE, sorry. So to basically the beginning of the Middle Ages or the early Middle Age period. Definitely Marek was already present somewhere in Europe by this point. And there is no mention in any historical records about these herpes viruses killing chickens in masses. So we can fairly assume that at this point it was probably at least less virulent than it is today. You can make a phylogenetic tree as Kevin showed before, and so here we have the best covered strains at the bottom, the ancient strains in green, which form a monophyletic clay that's outside of the variability of strains today that you find here in China and here in the West and the US, in Europe and the US basically. So it falls outside of the variability of the ancient strains. So what we found is basically kind of strains that don't exist today anymore. This is a circular version of so this is the genome that's a circular DNA virus here. Genome that annotated with the different genes across and some different annotation. I won't go into too much detail here, but this is the result of a lot of different analysis where we're basically comparing the genome of modern day virus and the ancient virus to try to figure out which part has evolved really fast. And which part is the most different, a bit like what Kevin was showing with the sheep and ancient sheep and modern sheep, or ancient goat and modern goats. What we can see particularly is that there is a peak of divergence. There's a part of the genome that's really divergent between the modern and the ancient that is overlapping with a gene that's called MEK, which is a gene that is an oncogene, and that needs to basically control the tumor development in the chickens that are affected by this disease. You can go into the sequence of that gene and try to figure out exactly how this gene has changed at the amino acid level, to try to figure out exactly what drives this molecularly, the virulence of these virulent strains. And so what we can see here is this is the strain this is the sequence of a modern MEK. And you can see here that these proline repeats here in the amino acid sequence. Basically what we could show is that the ancient Marek has many more amino acids, so many more proline repeats here. And so compared to the very virulent strains here, the VV plus MDV strain, which has only 2 proline repeats, and this is an ancient strain that would have 6 of these proline repeats. There's multiple evidence that suggests that this is really what's driving the virulence. So what we think is happening is multiple times these viruses have lost these proline repeats through mutations, and that has driven the virulence, or at least the oncogenicity of this virus. And then we did some lab experiments, some transfection experiment to try to see whether we could actually validate this in vitro. And so we assessed the transfection capability, transactivation capability, sorry, of this MEK gene of the ancient versus the modern MEK gene. And to make a long story short, we could show that basically when you have the transactivation capability of the ancient MEK gene are much lower than the transactivation capabilities of the very virulent and the mild strains that we find. So we could validate this in vitro. And so we are now working with colleagues in Berlin that are looking at doing some in vitro tests, sorry, in vivo tests to see whether this ancient virus could actually lead to could lead to a sustained immune response in the chicken while not leading to strong symptoms. So this is we're hoping that this can help us understand more about the biology. We'll definitely understand more of the biology of the virulence of this virus, but also maybe help us with creating treatments and maybe vaccines in the future. The other question that I was bringing, and I will just really quickly go on this and I'll stop, is that the other question that we had is just what drives the virulence molecularly, but where is Marek disease virus coming from, or how did it originate? And we find it about 1,000 years ago in Europe, but before this, we don't really know. One interesting thing though that we started looking at is that Marek disease virus has a telomerase RNA gene in its genome that it acquired from a host, and so this gene is basically used to delay senescence in a cancer cell, and so what you can do is you can grab the sequence of that gene in the Marek virus genome and compare it to a bunch of sequences of different species to try to figure out which species was it infecting before it infected chickens, if we can, or was it infecting chickens at first? And what's really good is that we have tons of data that we can compare this. So we can take the telomerase genes in the virus, and then we can compare it to we have about 5,000 chicken genomes from all over the world, including wild, domestic, and now there are hundreds of genomes from birds from all over the world, different families of birds. So we can then compare this sequence across all these data sets and try to figure out what is the best match for this. So where did this telomerase gene come from? And to make a long story short, we can then compare this, across the phylogeny of birds. Here, this is quite preliminary, so its figures are not that nice, and we can look at the sequence identity. And basically what we find is that this telomerase gene in the virus is most closely related to the sequence that you find in guinea fowl and not in chickens. And this is quite interesting because, it suggests that maybe it first was affecting guinea fowl and then jumped from host into chickens. And that brings us to the possibility that actually the Marek disease was actually acquired through Africa, to trade routes with Africa as we started to expand trade route as humans and started to bring animals, domestic animals, across the Sahara. And at some point where basically the guinea fowl and the chickens met. And we did some work about a few years ago with colleagues from archaeological colleagues, where we showed that really chickens don't only come in sub-Saharan Africa in the 10th century or maybe 9th century CE, so maybe 800, 900 years after BP. So no, sorry. 1,200 years BP, so before present. So we think that maybe it's possible that actually Marek only came into chickens once chickens and jungle fowl, and guinea fowl came in contact through this Indian Ocean trade that happened during the Middle Ages. And so we are looking into how we can test these hypotheses a little bit further. But yeah, we think this is getting quite interesting now. And then, so the virus came into Europe about 1,000 years ago, stayed there for a while without really raising any alarm bells, and then we started expanding our chicken industry, and then we created this sort of monstrosity as a result. All right, this is my lab. A lot of collaborators and people have provided samples and various help. So yeah, and I think there might be time for question. Thank you very much.
Perfect. Thank you very much, Laurent and Kevin. And just for anyone in the chat, if you have any questions, to just leave them in the Q&A box there at the bottom, and I can pass them on to both Kevin and Laurent to answer.
Yeah, I have to run for another presentation, so I'd just like to thank Laurent as well for presenting, and I could leave you maybe with a question, Laurent.
I can
Is there
answer
any
your questions too, Kevin, if you want, maybe.
I do. Unfortunately, I do actually have to run. I'm going to be late
It's
for this.
fine. No, go ahead.
But is there any hypothesis about that initial increase in disease frequency? Like, you know, is it just by chance in the early 20th century this just evolved, or was it an early consequence of early like upscale production? With that, I have to go.
Okay. I can answer Kevin's question. But we don't really know how prevalent it was in the past, but we assume it wasn't too prevalent, given we haven't found that many bones that have it. So if it was prevalent, it was probably not systemic, because we have a lot of chicken genomes from ancient sources, and the majority of them don't have any Marek virus. So we assume it's not that prevalent, but it's really hard to tell.
Yeah, I don't see any questions coming into the Q&A session here at the minute, which I'm going to take, given that you answered everything so well that there isn't any questions from any attendees. And yeah, I would just like to thank both Laurent and Kevin for a very interesting and insightful talk, and to all of the attendees as well for attending this. This video will be posted to YouTube as well, so we can watch back to get any further insights into things as well. And hopefully we'll see everyone again at the next UCD research seminar series. So again, thank you very much, Laurent.
Thank you.